Hello, everybody. My name is Sydney Nimtz, and I am going to be doing my first discussion on RNA interference. RNA interference is a biological process in which RNA molecules inhibit gene expression or translation by neutralizing targeted mRNA molecules. So for background information / introduction and principles of techniques, what I have here is RNA interference, or RNAi, is a technique first discovered in the nematode worm. The RNAi pathway is found amongst many eukaryotes. RNAi is most frequently used in order to inhibit as well as regulate gene expression and translation. In simpler terms, RNAi is a system that helps to control which genes are active and how active they are. RNAi is used mostly in functional genomics for the treatment of various disorders, infections, and cancers. Although RNAi is relatively newly discovered, it has scientists hopeful that RNAi will be a major key in the breakthrough of experimental biology. For the application of technique, RNA interference has two key components that allow this technique to be possible. The first is microRNA, or miRNA for short, and the second is small interfering RNA, or siRNA. Both of these small RNA molecules act in directing enzymes to degrade messenger RNA, as well as preventing translation. The pathway is controlled by the RNA-induced silencing complex, or RISC, and is initiated by the enzyme dicer, which cleaves the RNA into short fragments. RISC prevents the mRNA from being used as a translation template. All of this in turn gives biologists an idea of the specific function of that particular gene. For my figure from a primary research article, for A, about the author's question that they wanted to know, the authors of the figure I chose wanted to know if RNA interference could represent a new approach for the treatment of HIV infection. HUH7 cells were stained with an antibody to NS5B, which were stained in red, and benzimide, a nuclear stain, which was stained blue. What the figure is showing is 6 different pictures labeled A through F, where each shows us something different about the localization and expression of the S1179L cells. The figure suggests that based on the stains, RNA interference can block HCV replication in HUH7 cells, alluding to the conclusion that RNA interference could be a possible resolution to the treatment of the HIV infection. Some strengths I found in the experiment were that it was very detailed and specific, and some drawbacks that I thought could arise were that there was a lot of room in this experiment for human error to alter the overall results of the experiment. A question I would like answered about RNA interference is, how could RNA interference be used as a breakthrough resource for biologists trying to cure cancer? In a lot of the research that I was looking at, it mentioned multiple times how RNA interference could be a resolution for some cancer treatments, and I would just like to know from some of my classmates how they think RNA interference could play such a big role in this. Here are the list of my citations, and thank you all for watching. I had a really great time researching about this topic and learning more about it. Thank you.